Hi, everyone. My name is Peter Koo. I'm an assistant professor at Cold Spring Harbor Laboratory, and I'm excited to present work led by Shushan Tonyan on interpreting cis-regulatory interactions from large-scale deep neural networks for genomics. And transcriptional regulation requires a complex coordination of many proteins. If you look at this overly simplistic diagram, there are many proteins interacting with DNA at various cis-regulatory elements, also known as CREs. These CREs then go on to interact with each other to form enhancer promoter complexes. And this transcriptional regulation process is incredibly challenging to model mathematically because of so many moving parts and the dynamics of it. Problems like this exemplify why deep learning is such an attractive approach, because the deep learning model is a powerful function approximator. So in principle, it can be trained to take DNA sequences as input and predict the experimental readouts of these functional genomic assays that you see here. Now, these sequence of function deep learning models have been incredibly powerful tools to not only fit functional genomics data well, but also to interpret their underlying cis-regulatory mechanisms. From a function approximation standpoint, when training a deep neural network on functional genomic assays, we're essentially training it to learn the underlying function of that experimental assay. And so we can then use a trained model as a surrogate for that experiment, or as like an experimental simulator, where we can make hypothetical or counterfactual measurements for new sequences that the model hasn't seen during training. And this paradigm is incredibly powerful, as we can use a high-performing model to score the functional consequences of non-coding mutations. We can interpret the model to gain insights into cis-regulatory biology, as you'll see later in this talk. And we can also design novel sequences with desirable properties. But there's this big disclaimer here that this is all through the lens of the model, and it's not even all that clear whether we should trust their predictions. And in this is the part of the talk where I typically go into a long spiel about how we need to consider distribution shifts. But in today's talk, we'll simply assume that these neural network models fit genomics data well, and now we're just interested in understanding what they've learned. And to give you a little context, it's helpful to recognize that the trends right now in genomics are to build bigger models that consider longer sequence context and are trained on large troves of functional genomics data, and just like Informer and Borzoi. And so these models take in sequences that are hundreds of kilobases long, and they predict thousands of functional genomic assays. And it turns out, these Informer and Borzoi models are actually quite impressive if you consider their predictions for gene expression, via summary statistics across genes. Of course, they're not perfect, and they fail to capture many aspects of gene regulation. And this was highlighted in many follow-up studies where they compared Informer's ability to generalize to orthogonal experimental data sets. But this is currently the state of our field right now. All we can really do with these large-scale models is to see how well they can generalize to orthogonal experiments, and it doesn't really inform us of what these models have learned in a satisfying manner. Of course, we can perform attribution analysis, and that's what was done in the original study. And if you're not familiar with attribution analysis, it's basically like an in silico version of saturation mutagenesis, where we're measuring single nucleotide effects. And if you zoom into one region, you can see that Informer bases its predictions on patterns that resemble motifs. But at the scale of hundreds of kilobases, these low-level motif-level explanations are simply too complex for humans to process. So to address this, Shushan has developed CREM, Cis-Regulatory Element Model Explanations. CREM is an in silico perturbation toolkit to interpret large-scale models like Informer. And the basic idea is that we're not going to be performing perturbations at single nucleotide levels, but rather on extended DNA sequences at a CRE level, similar to what can be achieved with CRISPR interference technologies. But unlike CRISPR-I, all of our perturbations are done in silico, so we can really scale the numbers and the sizes of the perturbations that we're applying. But as the number of perturbations grows, the combinatoric perturbation design space grows exponentially, so it can be challenging to interpret the results of those perturbations. So what we've done with CREM is we designed a suite of different types of perturbation experiments from which you can take away precise claims about certain aspects of gene regulation. And I don't have time to cover them all today. I'm just going to highlight a few. And as a case study, we're going to interpret Informer, focusing on transcription start site activity, or TSS activity, in K562 cells using Informer's prediction of a CageSeq track. First, we ask the question, how does TSS activity depend on distal regulators? And to address this, we have the context dependence test. Here, we've centered our 200 kilobase sequences on a gene of interest, and we tiled the DNA into non-overlapping 5 kb bins. We performed a dinucleotide shuffle on the whole sequence while keeping the central TSS tile intact. And of course, we did multiple shuffles, in this case 100, and averaged the model's predictions. This randomization and averaging serves to marginalize out the contributions of all of those other contexts. And so this is effectively measuring how much TSS activity depends on just a proximal context within 5 kb of the TSS in the absence of any distal context. And we found that Informer exhibited a wide range of interesting responses. In many cases, shuffling the context led to a complete destroying of that TSS activity. This can happen if there are active enhancers that are really supporting that TSS's activity, and shuffling, of course, destroyed those enhancers. And so we refer to sequences with this response as enhancing context. And in other cases, shuffling the context had no effect on the TSS's activity, and so we refer to these as neutral contexts, and it should be no surprise that many of these were enriched for housekeeping genes. And in more rare cases, shuffling the context led to an increase in TSS activity, and this can happen when there are active silencers that are suppressing that TSS, and shuffling destroyed those. And so we refer to these as silencing contexts. And we performed this on this test on over 10,000 genes in active genes in K562, and now we're just going to subsample 200 sequences from each of these context categories for further exploration. So next, we asked whether a single Cree is sufficient to drive TSS activity. So the goal here is to gain a higher resolution view of enhancers and silencers, as opposed to what we're doing previously, just looking at the entire sequence context. So this is just like that context dependence test, except now we're including an additional tile, an additional 5 kb tile, which I'm referring to as a Cree here, in shuffled sequences. And so we measure the Cree effect by using the average predictions when both the TSS and Cree are present versus when just the TSS is present. And we did this for every single tile across every context, every sequence context. And you can see that for sequences that we categorize as enhancing contexts, most tiles don't have any effect on TSS activity, as you would expect, but most tiles had a very weak positive effect, which means that they act as a weak enhancer. Very few tiles had a very strong effect on TSS activity. And in neutral and silencing context, there was a more even distribution of weak enhancers and weak silencers. And so this tells you that a single tile really isn't sufficient to drive the observed TSS activity levels. And so this 5 kb tile size that I'm using is really arbitrary choice. So here I'm going to show you how we can use CREM to gain a higher resolution view of sequence elements within those 5 kb tiles using a nested greedy search. And so the idea here is if we have a 5 kb strong enhancer that we're interested in characterizing or fine mapping, we can create subtiles of that 5 kb tile. And then what we're going to do is introduce shuffle perturbations again at each subtile. And the goal here is to prune out all the unnecessary tiles. So by the end of the greedy search, we'll end up with just a set of necessary and sufficient subtiles that can explain the full 5 kb tile. And to make this search more efficient, we do at 2 different resolutions. First at a 500 base pair resolution, followed by a 50 base pair resolution among the surviving 500 base pair tiles. And for comparison, we also compared the ability to identify sequence elements using motif analysis, using extreme, as well as saliency analysis, which is this grad times input here. Here's a visual example, a qualitative example for one 5 kb putative enhancer for the MyADM gene. Motif analysis basically says there's a motif everywhere along this sequence, and that's not really that informative. Saliency analysis is placing emphasis all over the place as well, whereas CREM finds 4 focused regions of sequence elements that really can explain the full 5 kb tile's behavior. And of course, we can make this more quantitative and across multiple the 5 kb tiles. Here I'm applying the log fold over control, which is basically the sufficiency of the subtiles identified by these methods divided by the sufficiency of the full tile. So a value of 1 is basically where you want to be at, and you can see that CREM is able to prune out much more sequences and identify really compact subsets of sequence elements that really can define that full 5 kb tile behavior. And from those putative enhancers and silencers that are identified by CREM, we can look at their biochemical properties by comparing the epigenetic marks that are enriched in those elements using ENCODE tracks. And I'm not going to go over the details here, but by and large, we found that they largely have the right characteristic features of known enhancers and silencers. Now, I mentioned before that a single tile, 5 kb tile, isn't sufficient to drive the observed TSS activity levels. So multiple tiles are needed. So here I'm going to show you how we can identify sets of silencers or sets of enhancers with this higher order Cree interaction test, which is done by a different type of greedy search. And so if we're searching for sets of enhancers, first, we'll perform shuffle perturbations on each tile independently and identify the shuffle that drops the TSS activity the most. That means that this is the strongest enhancer for that individual perturbation. Then we'll fix that shuffle and then move on to the next round where we'll shuffle the remaining tiles and identify the next tile that drops TSS activity the most. And we'll continue this over and over again. And we found that it takes roughly about 4 to 5 tile perturbations, enhancers, to drop TSS activity well below 80% of its original TSS activity levels. And it was also interesting to note that silencing contexts and neutral contexts also contain large numbers of enhancers as well, but with much weaker effect sizes, never able to drive TSS activity towards 0. And we can also do this this greedy search for sets of silencers. And here, the objective is to identify the tile perturbations that maximally increase TSS activity now. And we find that in silencing contexts, silencers come in much greater numbers, but they have weaker effect sizes. And neutral and enhancing contexts also contain small numbers of silencers as well. So taken together, it really seems as though Informer has learned that every gene has numerous enhancers and numerous silencers, and the net effect of these is what leads to the observed TSS activity levels. Now, the previous greedy search only identifies sets of enhancers, but it doesn't tell us how those sets of enhancers interact. So we can gain insights into how they interact by comparing them to different types of interaction models. And so here we compare them against an additive model, additive effects models of enhancers. And we largely found that they behave additively on average, but we can stratify these results and we can find scenarios where certain enhancers for genes interact subadditively, i.e. redundantly, or superadditively. And we also compared these interactions with a multiplicative model, but I'm not showing you those results today. And so in conclusion, these multi-scale in silico perturbations are powerful tools to interpret targeted biological questions from these large-scale deep neural networks. Using CREM, we found Informer's prediction of TSS activity is based on integrating numerous enhancers and silencers that interact in complex ways. CREM can be used to design or prototype optimal experiments to be validated in the laboratory. And I really believe that we're only scratching the surface of these types of perturbation assays for efficient discoveries. Moving forward, we'd like to explore more of the motif level interactions that are taking place within enhancers that interact multiplicative or additively or subadditively, for instance. We'd also like to explore compatibility rules between why certain enhancers only interact with certain promoters, as well as infer interaction graphs of these Crees. And so again, I'd like to thank Shushan Tonyan for leading this study. CREM is a package that's available that you can pip install. We have open source. If you want to, if you want to customize it in any way that you want to explore, and we also have a read the docs. And I also want to mention that Jessica Zhou has a poster today at 12:20 to 2:20 on learning to predict model uncertainty in genomic deep learning through knowledge distillation. And I think this is a really powerful tool that you guys should all see, because in one of her experiments, I know a lot of people are interested in predicting single nucleotide variant effects using deep neural networks, and we've explored that. What we found that it's actually pretty easy to predict model uncertainty, and if you prioritize the predictions according to uncertainty, you can find that when the model is very certain, the prediction of single nucleotide variant effects is actually quite good. And when the model is uncertain, it's actually pretty poor. So of course that makes sense, but now we have a way of quantifying that. And so you should visit our poster to learn more about that. And I'm happy to take questions. Two questions.
Let's see if this works. All right. I'm not making it work. But I'll speak loudly. Oh, right. The online people can't hear me. Testing. Yay. Okay. Hello, online. Thank you for a really compelling talk. The negative regulation really caught my attention. So in studying Wnt signaling pathway and doing a lot of ChIP-seq and a lot of RNA-seq to try to figure out what is the most upregulated transcription factor, we found it was SP5. SP5 binds to the exact same motif as SP1, although we found there was a subset of SP1 binding sites, because SP1 binds ubiquitously, that SP5 uniquely bound to. It looked like it was one particular nucleotide in the middle of the motif. Now, SP5 turns off what SP1 turns on. So here you have the exact same motif. It has one nucleotide potentially distinguishing which sites will bind to SP5. And those are the sites that in the right context will act as negative sites under certain conditions. Yeah. So I wanted to describe that scenario to you. It's basically a negative feedback loop. What are your thoughts?
I think that's really cool to explore. We did some just anecdotal motif analysis on the enhancer sets that we found and the silencer sets that we found. And it was just it's very superficial because we're running out of space in our manuscript and our supplements are really long too. But this is something that we can follow up, that I would love to follow up with you to chat after, because basically our silencers, we found a lot of enrichment for heterochromatin, transcription factors, but we didn't really know how to explore it because there isn't a lot of studies done on silencers, not as much as for enhancers.
Yeah, it'd be really interesting to drill down on the same exact sites that have both positive and negative effects.
And there is that big disclaimer that this is all through the lens of Informer, and Informer is somewhat flawed in many ways. And so, but we can still explore and learn things that it's learned. But we should definitely chat after.
Hi, great talk. And speaking about issues with Informer, I mean, you and like Julian Gagneux and Sarah Mostafavi have looked into more detail how well this actually works, and that most of the impact seems to be really local and not spending like distributed across these very large input windows. So how does this fit together now with this working with permuting many regulatory regions, not for the core region, the 5 kb center region that I believe, but like figuring out what all these dependencies between these elements are? Exactly.
It's an excellent point. These models, like Informer, have extreme biases, and people have really only explored enhancers before, like Alex Carolus' work in Julian's lab, they found that they have this extreme bias of being close to the promoter. But in our study, we've actually found that they consider silencers at very, very long distances away. So it's something that we don't really understand. It might just be that there aren't enough examples in the data set for it to learn these long range enhancers. And actually, we also explored Borzoi, which considers 500 kilobase pair inputs. And we find that it does consider more enhancers that are more further away, but there are all these other weird things that pop up with that model too. And so, what I think is they're not exposed to enough genetic variation as mentioned by Carl's talk, and it's just a matter of how do we figure out what the optimal training sets are for these models to learn distal regulatory interactions. But I agree, these models right now are really good at modeling within cis-regulatory elements incredibly well. But like that's not all of gene regulation, and so we have a long way to go.